Welcome to my channel Biography Zone.
Discovery of lysozyme, a pioneering step toward antibiotics. Before Alexander Fleming revolutionized medicine with the discovery of penicillin, he made an earlier breakthrough that hinted at the existence of natural antibacterial agents within the human body. In 1921, while conducting routine bacteriological experiments at St. Mary's Hospital Medical School, Fleming stumbled upon an enzyme capable of breaking down bacterial cells. He named this enzyme lysozyme. Although lysozyme was not a powerful antibiotic, its discovery was a crucial milestone in microbiology. It demonstrated that the body had natural defenses against bacterial infections and reinforced Fleming's belief that bacteria could be selectively destroyed without harming human cells. This revelation laid the intellectual foundation for his later discovery of penicillin in 1928. The accidental discovery of lysozyme. Like many great scientific discoveries, the identification of lysozyme was largely accidental. Fleming was conducting routine experiments on bacteria, examining cultures under a microscope. His goal was to study how bacteria interacted with the human immune system and whether natural substances in bodily fluids had antibacterial properties. One day, while suffering from a mild cold, Fleming sneezed onto a bacterial culture plate. Instead of discarding the contaminated sample, he decided to observe what would happen. To his surprise, he noticed that the mucus had a dissolving effect on certain bacterial colonies. The bacteria in the areas where mucus had fallen appeared to be weaker or completely broken down. This observation intrigued him. What was causing this reaction? Was there a previously unknown antibacterial substance in the mucus? Fleming decided to investigate further. He carefully isolated and analyzed the affected areas of the bacterial cultures and identified an enzyme that was responsible for breaking down the bacteria. He named this enzyme lysozyme, derived from lysis, which means breaking down or disintegration, and enzyme, a biological catalyst. Expanding the research, where is lysozyme found? Realizing the significance of his discovery, Fleming began an extensive study of lysozyme, aiming to determine where it could be found and how it functioned. He quickly discovered that lysozyme was not limited to nasal mucus, it was also present in other bodily fluids. Through a series of experiments, he found that lysozyme was present in tears, saliva, sweat, breast milk, seminal fluid, egg whites. This discovery suggested that lysozyme was a naturally occurring antibacterial agent produced by the body to fight infections. Fleming hypothesized that it played a role in the body's first line of defense, helping to prevent bacterial infections before the immune system needed to respond. The presence of lysozyme in tears and saliva was particularly fascinating. Tears are constantly produced by the lacrimal glands, keeping the eyes moist and protected from environmental pathogens. Similarly, saliva contains enzymes that help break down food, but it also serves as a natural defense mechanism, preventing bacteria from growing in the mouth. How does lysozyme work? Fleming's research revealed that lysozyme functioned by breaking down the cell walls of certain bacteria, leading to their destruction. It specifically targeted peptidoglycan, a key component in bacterial cell walls. When lysozyme came into contact with susceptible bacteria, it caused the cell walls to weaken and rupture, ultimately killing the bacteria. This process is known as lysis, and it prevents bacterial infections from spreading in the body. However, Fleming quickly realized that lysozyme was not effective against all bacteria. It worked well against non-pathogenic or less harmful bacteria but was ineffective against more dangerous pathogens, such as Staphylococcus aureus and Streptococcus pneumoniae. These bacteria had thicker cell walls that were resistant to lysozyme's effects. Despite its limitations, the discovery of lysozyme was groundbreaking. It was the first time a scientist had identified a naturally occurring enzyme that could selectively destroy bacteria without harming human cells. Scientific impact of lysozyme. Although lysozyme was not the miracle cure for bacterial infections, its discovery had several important implications. 1. Confirmation of the body's natural defenses. A lysozyme provided scientific proof that the human body had built-in mechanisms to fight infections. This reinforced the idea that the immune system was not solely dependent on white blood cells but also relied on chemical agents to combat bacteria. 2. Inspiration for further research on antibacterial substances. A. Fleming's findings encouraged other scientists to explore naturally occurring antimicrobial agents, paving the way for the development of antibiotics. 3. Development of new medical applications. A. Lysozyme became an important tool in microbiology and medicine, particularly in food preservation, to prevent bacterial contamination. Pharmaceuticals, used in eye drops and nasal sprays. Medical research, as a model for studying bacterial cell walls. Although lysozyme did not revolutionize medicine the way penicillin did, it was an essential stepping stone that shaped Fleming's future research.